Yeah, many thanks. It's great to be back here again. This meeting grows every year and gets more and more exciting. Many thanks to the organizers for doing just a fantastic job. Really appreciate it. So today I want to tell you about insights into stem cell longevity, rejuvenation, and senescence from the study of diapause. So I think we can all appreciate that dietary interventions such as intermittent fasting or caloric restriction can have profound effects on health span and lifespan, extending lifespan in model organisms, decreasing age-related markers in humans. And this probably happens in a way that goes beyond just simply slowing down metabolic rate and may in fact involve rejuvenatory processes. So for example, fasting mimicking diets can stimulate stem cell renewal in various stem cell compartments, yet the mechanisms underlying this remain elusive. Now, we've been long interested in a biological phenomenon called diapause, which is an extreme state of fasting-induced quiescence and longevity, in which animals arrest in a hypometabolic state that are resistant to stress and geared towards survival. And this state is reversible, and upon recovery, refeeding, it stimulates regenerative, rejuvenatory processes. And it's not only limited to invertebrate species. Certain mammalian species, for example, can enter into embryonic diapause when they experience nutrient deprivation in utero. And even adult animals can enter into hypometabolic states such as torpor or hibernation when exposed to wintering or nutrient deprivation conditions. And even animals such as us that don't have explicit states of diapause can respond to fasting and feeding and remodel their cell fate and cell metabolism accordingly. And so particularly within stem cell compartments where you have quiescent and active cells, I like to think of ourselves as mosaics that are comprised of diapause-like quiescent cells and non-diapause active cells, and that this balance of activation and quiescence is essential to stem cell longevity, tissue homeostasis, and organismal lifespan. And where this goes awry, so for example, in cancer dormancy, cells can hijack these mechanisms to escape immune surveillance and chemotherapy, only to emerge later as full-blown cancers. So it also has disease relevance. So the worm C. elegans is a master of diapause states. It's very responsive to its environment. And in particular, perhaps the most famous diapause state is this alternate third larval stage called the dower. The dower larvae are incredibly stress-resistant and long-lived, and a genetic dissection of the dower led to the discovery of DAF2, which many of you may well know is the insulin receptor. Modest downregulation of this pathway can extend lifespan across taxa, and this is very much dependent on another conserved regulator, FOXO. We work in this field not only on insulin signaling, but also on vitamin D and bile acid signaling, on DAF12 and other associated components. And this regulates both dower and lifespan as well. But today, I want to tell you about another diapause state that's been very little studied called the adult reproductive diapause, or appropriately for this meeting, ARD. So these are animals that are fasted in a late larval stage. They enter into a mini adult form that are in this sleep-like quiescence. Here you can see they're in this adopt a sleep-like posture. They're about a tenth the size of an adult animal that's fed ad libitum, yet they have all the adult features you can imagine. And these animals are remarkable because I think they can tell us about what genes and mechanisms can promote resilience and regeneration in the adult. And we want to test the hypothesis that going through fasting and refeeding mechanisms can reprogram cellular fate. So this shows you what these animals look like. So normally under ad libitum, under ad libitum conditions, animals live about 20 days, but under the ARD, they live about 80 days, so there's about a threefold increase in lifespan, and this is even more remarkably extended in cooler environments, they can live sixfold longer. And during this time, in particular, the only adult stem cells are the germ cells, and they're quiescent in this state. And the soma begins to degrade over time, yet refeeding them, even out to 60 days, they completely rejuvenate their soma and germline, and they can give rise to substantial broods. And so there must be remarkable mechanisms for preservation of germline and soma that have been conserved in evolution. And this is not only limited to the germline, but also we see, for example, gaps in neurites that get repaired upon refeeding, and there's this massive regrowth of muscle and mitochondria upon recovery. So here you have cognition, you have frailty being overcome, and later I'll show you immunity. And so if you look at the transcriptional changes that happen during ad libitum aging, there are genes that go down, genes that go up, but when you refeed animals coming out of ARD, you see essentially a transcriptional reversal of these phenotypes. And if we measure the age of animals in ARD at the end of a long period, they look like they're aged, but upon refeeding, you can reverse transcriptional age. So it does seem like we have a model of age reversal of rejuvenatory processes. So naturally, the question arises, what are the activities that are responsible for sustaining survivorship during ARD, for the resilience of ARD, and what are the activities that are required for recovery? And through genetic screens, we identified HLH30 or TFEB as an essential component. So TFEB is mostly known as a conserved regulator of lysosome biogenesis, autophagy, and fat metabolism, and is required in a number of longevity pathways in C. elegans. And in the absence of HLH30, instead of living 80 or 90 days, animals live a mere 10 days. And there's very little effect under ad libitum conditions, so it seems quite specific for the fasting state. This is part of a signaling mechanism, since TFEB goes into the nucleus within 2 hours of ARD induction, and then this is transient, since after about 2 days it goes out of the nucleus, suggesting that there are epigenetic mechanisms that take over. And beyond that, animals spend down their fat reserves quite rapidly, and they shrink in size compared to controls. But most notably, they're unable to recover from ARD upon refeeding and produce progeny. There's a very dramatic reduction of reproductive competence. And this is very specific to ARD, since HLH30 mutants have virtually no effect on dower formation or survivorship. So we were really intrigued by this phenotype of the absence of germline competence, and we wanted to look at this more carefully, in particular looking at germline stem cells and the germline stem cell niche, shown here in purple and green respectively. And so this is work by Tim Noninger, a very talented graduate student in my lab, and one of the first things that he noted was that when he stained for DNA damage markers, that these accumulate in the HLH30 mutants in a very significant manner. Also, if you look at the germline stem cells, the nucleolus undergoes expansion compared to wild-type controls, and these cells in the mutants are unable to enter the cell cycle compared to wild-type. And at this point, a little light went off in our heads and made us think, well, maybe this is like cellular senescence in worms within the germline. And just to remind you, cellular senescence often entails DNA damage or telomere shortening or oncogene activation, results in nucleolar expansion, the rest of the cell cycle, mitochondrial dysfunction, SA beta gal expression, and the expression of the SAS. And so we decided to look at these features in C. elegans. And indeed, if we look at these HLH30 mutants, they have mitochondrial dysfunction, manifest as reduced mitochondrial sphericity, also reduced oxygen consumption, and preliminary evidence suggests also increased oxidative stress. They also have elevated levels of SA beta gal, and there are many transcriptional changes among them. There's an increase in innate immune response, which we interpret as an increase in the SASP. And in fact, there's significant overlap with the SEN-MYO set of senescent markers. So this is really exciting to us because it's really the first time, I would say, in a comprehensive way that we have found a novel senescent phenotype not noted before in worms, a very tractable genetic model. And so beyond this, at the organismal level, we also see that these HLH30 mutants have poor movement, and they have accelerated transcriptional age. And this is not only a feature of this mutant phenotype, we also see this in wild type in animals that have been held for prolonged times in ARD. They're unable to enter the cell cycle. They also show nucleolar expansion. And so naturally we wondered, well, what are the downstream mechanisms that HLH30 TFEB is turning on that is important for this resilience and recovery? And naturally we looked at autophagy as one of those outputs, and we're very surprised to see that a number of different knockouts of autophagy genes had very little effect either on survivorship or recovery. And this was really quite perplexing. And so we decided instead to do a genetic screen, and this is a kind of a screen that directly selects for longevity in the following way. We mutagenize these HLH30 animals, and then we ask them to live longer and also recover and produce progeny. And consistent with this being a longevity screen, some of the mutations that we got out included those that reduce insulin IGF signaling, but also mutations in the TGF-beta pathway as well as cyclic GMP signaling. And so this just shows you some of the data. In red here is HLH30. It doesn't live very long, but if we now mutate DAF1, which is the TGF-beta type 1 receptor, we extend survivorship. Similarly, we extend survivorship by mutating the insulin receptor. Both mutations can recover a normal body size, and most dramatically, you can recover progeny production. Now, we decided to focus on this TGF-beta pathway because this was less well explored in the context of aging and also had a more profound effect on reproductive competence. And we then asked, well, what happens to these senescent phenotypes when we further mutate this TGF-beta signaling pathway? And indeed, we see that this prevents DNA damage, it prevents nucleolar expansion, and it restores the ability to enter into the cell cycle. It also reduces SA-beta-gal expression, reduces transcriptional age, and enhances body movement once again. And if you look at the transcriptome, there's a massive reversal of many of the transcriptional changes induced by HLH30 by further mutation of DAF1. Most notably, there's a reversal of the SAS phenotype, the innate immune signaling, which is normally upregulated in HLH30, is then downregulated by further mutation of the TGF-beta pathway. And so these genetic interactions suggest a model whereby HLH30 TFEB normally downregulates TGF-beta signaling to promote stem cell quiescence and resilience. And conversely, if we mutate HLH30, we'd expect that expression of these components would inappropriately increase. And so we decided to test this idea by using transgenic reporters, and I'm not going to go through all the details here, except to note that mutation of HLH30 can increase TGF-beta expression in neurons. It increases the expression of the receptor in the germ stem cell niche. And downstream from that, it also upregulates Notch signaling in the niche and within the germ stem cells themselves. Those are, Notch signaling is the ultimate regulator of the germ cell activity. And so this leads to the following model, that under fasting, TFEB goes into the nucleus, it downregulates TGF-beta and Notch signaling. This results in a quiescent, resilient state. And upon refeeding, TFEB goes out of the nucleus, and you get derepression of these growth signaling pathways and activation. And under a fasting condition where TFEB is knocked out, you get an inappropriate upregulation of these growth signaling pathways, and there's a disconnect between the nutrient supply and the growth signaling, which leads to macromolecular damage and senescence. So we think this is a really profound concept, because I think this happens more often than not within our own bodies with aging, that there's a disconnect between our nutrient supply and growth signaling, and this can lead to macromolecular damage. Just as food for thought, Aunt Simon has shown, for example, that human PBNC's TFEB goes down with age, suggesting that its absence could induce some of the immunosenescence that's seen within us. And it's also well known that TGF-beta signaling is part of the SASP, and maybe not just part of the SASP, but maybe instructive within the SASP. So perhaps some of the same signaling molecules are playing a role in immunosenescence. It's also been shown that TFEB and TGF-beta signaling are important for neural stem cell quiescence, and our work suggests that these two pathways might come together in a unified pathway. Finally, we asked the question, do these molecules play analogous roles in embryonic diapause and mammalian embryonic diapause, as well as cancer dormancy? And we had the privilege of collaborating with Manuel Serrano and Valentino Ramponi here, and they were actually doing CRISPR screens looking for guide RNAs that could affect the survivorship of cells induced into diapause using INC128, this PI3 kinase mTOR inhibitor, and they found that guide RNAs for TFEB were depleted from such cells, and if you knock down TFEB by siRNA, you reduce survivorship. Similarly, in human melanoma cells, if you knock down TFEB by siRNA, you reduce survivorship. So similar to worms, diapause and dormant states depend very much on TFEB. And similar to worms, you also see an induction of TFEB upon entry into diapause, as well as regulation of the TGF-beta signaling pathway. So I think it still remains to be seen whether there's direct regulation here, but the hints are there. And so we're very much excited to continue screening for genes that impact ARD recovery and survivorship, and we're continuing to find molecules that can, for example, whose knockdown can promote recovery from ARD or impede it. And one of the molecules that we found is very exciting. It's an epigenetic factor whose knockdown during the recovery phase, during refeeding, can actually accelerate growth. And it also improves muscle regrowth, and as well as body movement. So I think this is really exciting to us. On the other hand, if you knock out this gene during ARD, it reduces survivorship and it reduces recovery. It's actually expressed during the fasting state and turned off during the fed state. And so our model is that this epigenetic factor promotes the diapause state and prevents recovery, and that in order to have an appropriate recovery process, you have to downregulate this molecule. And this actually turns out to be a target of TFEB. So now we have something that's involved in the switching-like state. Tom Rando had mentioned earlier in the meeting how there's a trade-off between resilience and activation, and we think this is a molecule that could play such a role. And the sharpness of the boundaries between activation and resilience are very important to maintain. It's maybe not so much a trade-off, but really keeping the sharpness of boundaries between the two different states that may be important for a healthy lifespan and healthy stem cell longevity. And so with that, I'd like to summarize. I hope I've convinced you that diapause is amazing. It can yield key insights into longevity and rejuvenation from cell to organism, that this axis is a primordial regulator of stem cell longevity in diapause across evolution, that TFEB TGF-beta can link nutrient to growth signaling through a systemic mechanism and thereby prevent macromolecular damage and senescence under nutrient stress. And we think that fasting refeeding induces metabolic flexibility and thereby affects cellular plasticity required for reprogramming, perhaps not unlike the Yamanaka factors, and that ARD can reveal novel conserved pathways regulating adult rejuvenation and longevity. And so with that, I'd like to thank all the people who contributed to this work, my fantastic lab, our collaborators, sources of funding, and you for your attention.
Thank you. Thank you so much, Adam. That was amazing. I find myself in need of ARDD recovery every evening. We have time for maybe one or two questions before the photo.
Can you hear me? Do we know, in terms of the length of the fasting, how does it translate to every species lifespan? Let's say in human, how long the fasting has to be for us to see these biologic changes?
Yeah, that's a good question. I mean, in humans, for example, you don't see that many changes out to about 3 days. It's only when you take them on a long-term fast out to 7 days that you see these really profound changes. Now, the worm has been selected for an evolution to be able to outlast very long fasts because it never knows where its food is coming next. So, humans can also fast for 80 days. You couldn't just refeed them and hope for the best. I think they have to be gradually refed. But I think there are parallels, and we can identify similar pathways and mechanisms, even if the kinetics are slightly different.
Okay, last question for Rafa.
Adam, in terms of the signaling that is involved in the fasting response, there is a component that is a neuroendocrine stress response that is super important for the effect of calorie restriction in mice and in humans. How is that signaling in terms of the neurons in the worm?
In terms of the neurons, you said?
Right.
Good question. So what we do know is that the TGF-beta molecule is expressed only in a handful of neurons, which is equivalent to the worm hypothalamus. And that's where we see TFEB regulating its expression. So it's connecting sensory input through sensory neurons that are detecting food cues, regulating the nuclear localization of TFEB and thereby TGF-beta signaling. We also think insulin signaling is regulated within those neurons, although we have yet to show that in situ. Yeah, I think it's quite similar. Yeah.
Okay. All right. Thank you so much, Adam. That was really amazing.
Thank you.